I think it's time to start this event. Good afternoon, ladies and gentlemen. Today, on behalf of EFSA, I would like to welcome you to the info session on the EFSA's draft guidance on protocol development. Next slide, please. I'm Claudia Roncancio. I'm the head of the Methodology and Scientific Support Unit, and I'm here today with a group of colleagues and experts to provide some information regarding this document. Some practical information: you are now connected to the broadcast. The event is taking place in English and is going to be recorded, and it will be placed in the EFSA website together with the presentations. It's important that you provide us some feedback on the event. For this reason, we were going to submit you a survey. The deadline is next Tuesday, 4th of April, to get your feedback. Next slide, please. EFSA has been working on the protocol since some years, as you may know. Today, we have the opportunity to provide information on the draft document. This allows us to better explain the scope and the structure of the document, to enhance understanding, and of course to maximize the possibility to receive comments. Please note that concurrently this event, the public consultation is taking place. It will run until the 15th of May. The second objective of this info session is to address questions during the meeting, and this will be explained in a little while how to do it. The next slide, please. The guidance has been developed by a group of experts and has been also endorsed at the last scientific committee plenary in last February. Here you can see the number of colleagues and experts that have been taking place on this event, highlighted those colleagues and experts that are also intervening during this meeting. And if we go to the next slide, please, we can see a representation. The next one, please. Yes, we can see here the number of people that is joining this meeting. So we have 128 people registered. According to the affiliation, we have an important number of part of the audience from the university, public and research institutes, private sector, and the EFSA panel member and networks. Then we have also participants from the EU institution agencies, national authorities, and international organizations. I would like to thank all the colleagues and all the experts for your support and participation. I would like to thank particularly to Elisa Ayasa and to Spela Super for their support. And at this point, I want to give the floor to Thor Haldersen. He is from the University of Iceland, who will show us the agenda and the rest of the session. Please, Thor.
Thank you very much and welcome everyone. I will be chairing this info session here today and giving some background on the info session and on the document before we start. And as you can see here before you, the agenda here today is that in the first round I will give some introduction and background to the current guidance document, and this will then be followed by a presentation on the first session on the dynamic approach to protocol development at EFSA, the steps in protocol development, and a short introduction to the problem formulation. This will be followed by a round of questions, and after the coffee break we will move on to continue with the problem formulation, going in more detail into the sort of a harmonized framework which is suggested to be used in the guidance on formulating questions and sub-questions. And this will be followed by a second round of Q&A. And in the last section, we will give a presentation on the method specification on the protocol development, followed by some conclusions and recommendation, and the last round of discussion. Next slide, please. Now, we welcome any question that you may have that can help clarify the content and scope of the guidance documents, and any questions that may arise during the info session. And we would like you to submit your question in English via the Q&A chat. And preferably the questions that arise during the presentation, please state them as soon as possible so we can address them in the subsequent Q&A session. And it would be good if you could tag the relevant speakers using the @ sign so we can direct the question to the relevant person, but if not, we will allocate the question to the appropriate person ourselves. And those questions that we will not be able to address either due to time or other reasons, they will be answered in writing and published on EFSA's webpage by Thursday, the 4th of April at latest. Now, the questions that you may come up with here and during the info session, we will clarify, but any questions, any comments on the guidance document itself will have to be submitted formally through the relevant website. Next slide, please. And then a short introduction about the current guidance document and protocol development. Next slide, please. So it was in 2014 to 2018 that EFSA set out a series of principles which were aimed at increasing the rigor of its scientific assessment process. And these principles were very much focused on impartiality, methodological rigor, transparency, and engagement. And in line with these principles, the need for sort of using protocols and developing protocols prior to conducting scientific assessment was identified, and the need for a guidance on that issue was also identified. And the initial output was a technical report that was published in 2020, which provided recommendations for protocol development. It is equivalent to a guidance, but it was not a formal document, so it is a technical report. And that is what you see here on the right side. Now, what was already envisaged when the technical report was published in 2020 was the need for formal guidance. And the reason why the approach taken with the technical report first was that given the broad scope of EFSA scientific assessment and with a new methodology being introduced in terms of protocols, there was a need to draw on experience from different panels. And to do that, information on the technical report and how the use of the technical report and the guidance provided there was drawn from or collected between 2020 and 2021, and which helped us develop the current guidance. Now, secondly, there was also the need for a structured framework and guidance on problem formulation was also identified, and that was not covered extensively in the technical report from 2020. And we already knew at the time that such a framework on problem formulation should follow a harmonized approach that could be adapted to various different EFSA domains, because what we are dealing with in EFSA are scientific assessments that are quite different and have a rather broad scope. Next slide, please. Now, in terms of the terms of reference that we received for developing the current guidance was to fine-tune the draft recommendations that had been published in the 2020 technical report and translate them into a formal guidance document for protocol development. And we should keep in mind that the guidance should consider the feedback and the proposals that had been collected during the testing phase of the technical report, and an appropriate framework for problem formulation should be included. Now, what we also knew at the time, but became more and more aware of, is that the guidance should ensure flexibility and enhance harmonizations across EFSA's areas. Next slide, please. So the applicability and scope of the current guidance, the aim was to provide all EFSA panels and units with a harmonized and flexible framework for developing protocols for generic mandate. And the flexibility is quite important because sometimes we have quite comprehensive assessments and sometimes we have rather short assessments, and the flexibility needed to take into considerations the various scenarios that we can be in. Now, we realized also that the guidance may not be needed for scientific assessments which are covered by existing regulatory framework for scientific assessment. And to accommodate different requirements or mandates across EFSA domains, the guidance proposes a dynamic approach to protocol development for two main steps in the development. That's the problem formulation and the specification of methods. Next slide, please. And in terms of the process for developing guidance, next slide. During the testing phase, there were 21 different EFSA mandates where protocols were developed, and we could draw on the experience from those development. And the lessons learned was the need to increase user friendliness of any of the new guidance. So be basically clear and simple, to have more examples so people could adapt and relate to what they were doing, and explanations on how to translate mandates into scientific answerable questions and sub-questions, which is the problem formulation. Now, I think from my side, the most important, or one of the more important aspects of this work is that we were working together across various different EFSA domains, experts with various different backgrounds, and the work really highlighted the similarities and differences across panels, and that provided a very important feedback into the guidance that we are presenting today. Next slide, please. And on the problem formulation, the guidance on, or sort of the theoretical fundament for that part of the guidance was based on outsourced project with the aim to develop framework for problem formulation, which was done by Risk Science International. And during that work, they actually went over 700 EFSA scientific assessments, reviewed them, and sort of compared similarities and differences and how they would map these mandates into different assessment questions and sub-questions. And there we could map the similarities and differences across domain. And through this activity, a harmonized approach for formulating assessment questions and sub-questions was put forward, with what we call the a priori paradigm, which we would go through in the second round today. Now, concerning the guidance is complemented by a separate supporting document that acts as a template for protocols. It is the Annex E of the guidance, and it guides the user step by step through the process of planning and EFSA scientific assessment. So the guidance document is two, so more or less the guidance document itself, the document, and then the annex. And now I would like to give the floor to Elisa Ayasa, who will talk about the dynamic approach to protocol development. Thank you.
Thank you, Tor, and good afternoon, good morning, everyone. So I will start summarizing sections 3 and 4 of this draft guidance. First of all, the scientific assessment process at EFSA. EFSA Strategy 2027 and EFSA's quality management system set out the requirement for EFSA generic mandates for being conducted through a two-phase approach. And this approach consists of a planning phase, the so-called protocol development, and the implementation phase. Protocol development implies problem formulation. We will see it clearly throughout this info session. So the definition of the what the assessment aims to achieve, and the specification of the methods for the assessment. So the definition of the how. The implementation phase consists of implementing the protocol, verifying the compliance between what had been planned in the protocol and how then things were done, and documenting and reporting the entire process. This two-phase approach is a requirement for EFSA's so-called generic mandates, that are the mandates not related to processes of regulated products. Next, please. With regard to these assessments, the ones related to regulated products, the protocol, so the planning phase, is already covered by the relevant legislative framework and also scientific framework, which typically summarize the data requirements for a dossier and the methods for dealing with those data. So, as mentioned by Tor already, our guidance, this guidance is meant for generic mandates. It might turn to be useful for the cases when the regulatory framework for assessments of regulated products is not extremely detailed. Next, please. What are the advantages of planning upfront? Well, it is well established that defining the methods for a scientific assessment a priori before starting the assessment itself helps reduce so-called result-driven methodological decisions. Take, for instance, the case when one could be prone to include a study because the results of the study are already known. Or it also helps safeguard against the subjective decision-making throughout the assessment process because the plan is already done, as well as it protects from sometimes types of bias, like, for instance, the tendency to focus on evidence that is in some way in line with expectations. Planning upfront also helps increase transparency for sure, and for highly detailed protocols also helps streamline the scientific assessment process. So protocol development is well established in primary research, in systematic review, take for instance the approach of the Cochrane collaboration, and it's been applied as extended to various areas and many organizations that conduct assessments similar to EFSA. So here we have a couple of examples, for instance, from the US EPA and WHO. Next, please. EFSA works in a so-called mandate-driven environment. So EFSA produces scientific advice for regulatory purposes. So it's important to highlight here that EFSA assessments must be fit for purpose, on time, and efficient. What does it mean when it comes to protocol, to planning? Well, the plan for the scientific assessment must be clarified with the mandate requester before the start of the scientific assessment process, and, very important, adapted to the specific characteristics of each mandate. What does it mean in practice? Well, for protocols, this implies defining and tailoring the extent of planning of the protocol, the degree of detail of the protocol, but also the approach to disseminating and publishing the EFSA protocol. With regard to the first, so the extent of planning, the degree of details in EFSA protocols, this, depending on the characteristics of the mandate, can really change depending on the situation, and that's why EFSA protocols can vary according to this guidance, from being outlines of the problem that is at hand, the questions and sub-questions that must be answered, as well as of the overall approach that will be applied, to very detailed, extensive plans for the assessment that illustrate, for instance, the eligibility criteria for study selection, the approach to appraising, and so on. In addition to adapting the protocol to the specific requirements and characteristics of the mandate, there can be situations, reasons why it is not always possible to have fully detailed protocols. That is, for instance, when it is not possible to fully formulate the problem a priori before starting the assessment. This is the case, for instance, of adverse outcome pathway development, where the definition of the adverse outcome pathway per se is part of the assessment process and therefore cannot be fully detailed a priori. Additional reasons for not being able to planning in detail upfront could be, for instance, limited knowledge of the evidence out there, making it impossible to plan a priori, for instance, the approaches to appraising the evidence, or unavailability of methods. With regard to the second aspect that must be modulated in EFSA protocols, so the approach to disseminating and publishing, that can be different situations. So the case when the final protocol is published before the start of the scientific assessment process, at the end of the scientific assessment process together with the final output, or also the case when EFSA publishes draft versions of a protocol in order to seek feedback and input from the public, for external stakeholders, before then finalizing the protocol. So all these aspects are modulated in EFSA protocols. Next, please. Now, the last part of my presentation, what are the steps in protocol development according to this guidance that we are presenting today? I'm going to outline these steps because this will be the core of this info session and will be thoroughly described by the next speakers. So two main steps in protocol development, we've mentioned it already according to this guidance. Problem formulation, the definition of the what, and the specification then, step 2, of the evidence needs and the methods that will be applied. Each of these steps consists of multiple sub-steps, not all necessarily mandatory and needed, depending on the situations. Problem formulation. For sure, in EFSA assessments, problem formulation consists of translating our mandate terms of reference into as many assessment questions and sub-questions, and we will look at this in the next presentations, as needed. Formulating these assessment questions and sub-questions in a way to make them answerable through the scientific assessment process, and this guidance provides a new paradigm for this process. defining the conceptual model, the relationship among the assessment questions and sub-questions, and then, if needed, determining the relative priority of the questions, defining the overall approach as to whether it's going to be quantitative, qualitative, semi-quantitative, and also for highly detailed protocols, documenting the methods applied for problem formulation. With regard to the second step of protocol development, the definition, the specification of the methods, well, this implies defining the methods for answering so-called individual sub-questions, as well as the methods for answering big questions. With regard to the first, individual sub-questions, the guidance illustrates 5 possible methods. I'm not going to give the details of these methods here because they will be thoroughly illustrated at the end of this info session, but there can be 5. With regard to the approach to answering big questions, that can be these 2 situations when evidence is integrated across sub-questions. Take, for instance, the case of hazard identification and characterization in chemical risk assessment, when we integrate evidence across sub-questions and evidence lines, or the case when individual sub-questions are answered in a logical sequence in order to get to an answer to the final big question. Uncertainty analysis is inherent in this process, and ideally, although very challenging, should be planned for each and every step of this process, so in highly detailed protocols. Just a few points on this process, and this is my last slide, the next. This process, the process for protocol development, you may go to the next slide, please. Yes, thank you. This process starts after the acceptance of the mandate, after this clarification with the mandate requester. However, it's an iterative process, protocol development, which may require multiple consultations with the mandate requester in order to ensure the terms of reference are properly targeted and the approach is adequate. It may also require multiple sets of literature scoping, as well as consultations within the expert group responsible for the development of the protocol. Especially for highly detailed protocols, this process can be very time and resource intense, and for sure requires multidisciplinary expertise. So the guidance outlines the importance for EFSA protocols for the time dedicated to the EFSA protocols to be proportional to the overall time dedicated and assigned to a mandate. And with this, I finished with my presentation, and I'm giving the floor to Laura Martino for the next presentation. Thank you.
Thank you, Elisa. Thank you, Elisa, and good morning, good afternoon, everybody. In this presentation, we are going to explore better the content of the guidance as for the problem formulation. That's, as anticipated by Elisa, is one important step in the protocol development. And you see displayed here an overview of the steps of the process that have been already listed by Elisa. So first of all, we want to translate the terms of reference of the mandate into a series of assessment questions that need them to be operationalized, in the sense that we want to clarify, formulate them accurately in a way that can be answered with our scientific assessment process. Then in many cases, we would also need to break down these assessment questions, to split them in specific sub-questions that again need to be operationalized, so clarified, formulated in a way that helps us then to define the evidence needs and the methods to be planned in the protocol. And linked to this also, importantly, we need to define the relationship across questions and sub-questions. Looking at the right-hand side of this figure, you see the output of these first 2 steps. That is the definition of the scope of the assessment, the list of the operationalized questions and sub-questions, and the conceptual model. And you see also that actually this process is quite iterative, in the sense that it might require several rounds of discussion, not only within the group of experts, but also with the mandate requester, because an important aspect of the problem formulation is that to define questions, a sub-question in a way that can address well the request or the mandate requester. So at the end, the mandate must be fit for purpose. Moving along, sorry, the previous still. So moving along the steps of the problem formulation, you see that in step 3, we determine the relative priority of the assessment question and sub-questions. And we will see later on which criteria can be used for this exercise. Then we have 2 optional steps that are the selection of the overall approach for the mandate that might include, for example, to define whether we want to have a qualitative, quantitative, semi-quantitative approach. And again, the optional documentation of the methods that been applied for the problem formulation. Next, please. So moving to the first 2 steps that are very key in problem formulation, because they really deal with the translation of the mandates into the list of questions and sub-questions that we are going to plan in the protocol, and the definition of the relationship across them. The guidance discuss these 2 steps in the sections from 6.1 to 6.3, and stress the importance of a series of methods that can be used for this, for achieving this objective of defining question and sub-question. And those methods include, first of all, the scan of the scientific literature that can be very useful to identify, to capture elements that might be critical in the formulation of the question, and also to define the scope of the entire assessment, because having a look at the scientific literature out there, we can have a better idea on what is available, what could be critical issues for the mandate. Of course, we cannot go without the knowledge of the experts that need to assure that the formulation of the question is sensible from a scientific viewpoint. And again, it is meeting the expectation, the request of the mandate requester. The APRIO framework that we recently developed, that we are going to discuss more in detail after the coffee break. And last but not least, the conceptual model that is very useful since it can help displaying all the list of questions and sub-questions, and also help us understanding whether we need to refine them, how they relate, and yeah, to whether we need to refine them further. Next, please. So the translation of the mandate into the assessment question and sub-question. Of course, this is a key step, as we said. And normally this is necessary in EFSA, particularly the step of breaking down the main assessment question into finest sub-question, because we normally receive mandates that are quite complex and broad. And you can have an idea on how this process can go on, looking at the right-hand side of the slide. For instance, here we have an assessment question that is dealing with the health risk in the human population related to the exposure to chlorinated paraffin. And you could have a first tier of sub-questions that reflect the traditional pillars of effect identification, characterization, exposure assessment, and risk characterization. But then, as you can see, for each of those, you may need to have a further breakdown. For instance, for the effect identification, there is quite a long list, 7 sub-questions at the level of tier 2. And you could go even farther, so develop even tier 3, tier 4, whenever it is necessary. And at the highest level of granularity, a sub-question theoretically is something that could be answered by a single study or a single question in an expert elicitation process. And the question you might pose is whether to stop in this breakdown into sub-question, and there is not one size fits all for this type of challenge, in the sense that it really depends on the type of evidence attends to the specific needs of the mandate. So this is really something that need to fit the specific requirements of each assessment. And again, this process is very iterative, in the sense that going down to tier 2 sub-questions definition, you might need to go back at higher level, so tier 1, to further refine the definition of this question. Next, please. The conceptual model is another essential part of the problem formulation, since it clarifies the relationship across assessment question and sub-questions. And the conceptual model can take the form of a graphical display, but can be also a tabular, can have a tabular format, or can be simply a narrative description of the list of questions and their relationship. And very important is that it helps planning in the protocol, the process for answering the assessment questions. It really leads in the process or on how to combine the sub-question to come up at the end to the answer to the assessment question and the entire assessment. And you see below 3 examples of conceptual model. The first one is when actually the sub-questions at a certain tier level need to be integrated in order to address the higher tier question. And this is really the example that was provided in the previous slide. But conceptual model can also be used to show how sub-question can be used in a logical sequence. You could have, for instance, that depending on the reply that you provide at the level of tier 1 sub-question, you might decide to go for one choice or another. So in practical terms, the conceptual model will be a sort of decision tree to guide across the assessment process. And you might also have a conceptual model that describes the pathway to harm or to benefit even. And in this case, it will be really describing a series of events that leads from the initial exposure to the agent up to the outcome and the output. Of course, there may be also other types of conceptual model that we haven't thought about that are still possible. Next, please. So the third step, as anticipated, is the determination of the relative priority of the assessment questions sub-question. This is described in the section 6.4 of the guidance, where we indicate a series of criteria that could be used in order to perform this process. And the first obvious criterion is whether the assessment question or sub-question has a strong influence on the overall conclusions. Of course, the stronger the influence, the higher is the priority. But the decision of prioritization could be based on also on the fact that maybe one question or sub-question may appear in several terms of reference of the mandate, or in addition, it could be due to the level of novelty of some elements that appear in the question or sub-question, or the level of novelty in the evidence that we are going to use in one of these sub-questions. Last but not least, a criteria could be also the scientific controversy regarding this, the content of the sub-question that could require a deep investigation, for instance, of the level of uncertainty related to the evidence and the methods that he used. And very important is to document carefully how the decision on the prioritization has been done. And examples for these are provided in Annex A to the guidance. Next, please. So linked to the prioritization is also the possibility that doing this exercise, we identify sub-questions that we want to scope out. And this could be done, for instance, because we are focusing on a pathway for that specific sub-question, a pathway of exposure, for instance, that although it is plausible, is not expected to have a strong influence on the overall exposure. Or could be that we are dealing with an intervention, a measure that can be potentially effective, but as compared to other possible intervention is expected to be less effective. Think, for instance, of a series of measures that are defined to prevent the introduction, I don't know, of avian flu in the poultry farmhouse. You can start with a long list of possible measures, but then you identify the ones that you expect upfront to be more effective. And again, very important to document in detail the rationale for excluding sub-questions that should be indeed very carefully documented and reviewed. Next, please. So the fourth step is optional. It is not covered in a specific session of the guidance. It is about selecting the overall approach. It is described in the introduction for the problem formulation. And basically, this overall approach can imply a discussion about whether we want to use a qualitative, quantitative, or semi-quantitative approach, but also whether we want to adopt a tiered approach, for instance, whether we want to start from estimates that are less accurate but more conservative, and then we move progressively towards more accurate estimates. And if feasible, also as part of this overall approach, we could describe what are the resources and the timelines attached to each question and sub-question. Next, please. The last step is documentation of the method applied for the problem formulation. This is also optional, and it is not covered in any specific session of the guidance. Whenever possible, of course, for transparency, it is always useful to do so, but in the guidance, this is described as something that, although it is, let's say, advisable, it is not mandatory. And again, also the level of detail that we can put in this documenting the method for problem formulation is left to the working group. And this is my last slide, so I hand over to Thor.
Thank you very much. And now we have arrived a bit early to the first round of questions and answers. So I would like to invite people online to put forward any questions that you have on this first part of the info session, or any relevant questions that you have on the guidance related to what we have been going through. And Yeah, and if nobody has a question, I would maybe like to ask Alicia about sort of during the development of protocols and so on, what is the sort of the experience in EFSA now in terms of publishing protocols up front, which has been done in some cases, and in the other cases they are published after the opinion is finished. So what is your sort of experience with that process? Has it been?
Yes, thank you, Thor. Yes, indeed, there have been quite some experiences so far here at EFSA on publishing draft versions of the protocols, even holding workshops on these draft versions of the protocols in order to fine-tune them and ensure the methods were properly reported, indicated in the protocol. So there had been the experience, for instance, of the sugars opinion in the past, the sweeteners more recently, but also the BPA and quite some others. So for cases when at the start of the scientific assessment process, when the mandate arrives, the need for extensive consultations are indicated, EFSA follows this approach. As I said, it really depends on the type of mandate and the requirements for the specific mandate, because of course these consultations take time and resources, so it might not be always necessary to have them. Yes,
Okay,
thank
and we
you.
have a question from the audience. So do you expect that this protocol will influence the way the European Commission will give mandates to EFSA? And maybe Laura, if you would answer that one. Muted.
Sorry, I was muted. I would suggest that Claudia answer this question.
Okay. Claudia. Maybe Elisa, if you could answer that.
Yes, I hope. Wait. Okay. Well, for you said if it influences the way the mandate requester poses questions to EFSA.
Yeah.
Well, for sure, EFSA is, and this is also outlined in EFSA's quality policy, which was published, adopted and published back in January last year. For sure, EFSA is now trying to implement this approach that implies this clarification with the mandate requester when the mandate arrives on the so-called scientific value for our outputs, which covers multiple aspects of the final output, including the extent of planning in the protocol, the approach to publishing, etc. So maybe rather than influencing the way the mandates are posed, it will definitively affect this process of clarification between the mandate requester and EFSA, and in order to ensure that the protocol is tailored to the needs of the mandate.
So basically, in summary, it will not necessarily influence the way that the mandate is given, but it would facilitate dialogue so you have a more clear mandate and better. Yeah, thanks. And there's another question. How does the concept relate to specific protection goals? That's question number 2, and maybe Laura, if you could comment on that.
Yeah, I think, the guidance applies equally. Sorry, I don't know whether you hear me now. The guidance applies equally to any generic mandate So also to this case that was mentioned, there is not, it is very cross-cutting, as you can see also from the content of the guidance, but also from the templates that we provide in the Annex A. So there is not specific reason why it shouldn't apply to any domain.
Yeah, thank you. And there's another question. It has been indicated that the guidance in principle applies to generic mandates, but might apply to regulated products as well. Could you provide more information on when, how that might be the case? And Elisa, if you could comment on that.
Yes, indeed, because as we said, the guidance is meant for generic mandates, so mandates that are not related to processes of regulated products. However, there can be areas of EFSA and EFSA's assessments of regulated products where the legislative framework and scientific guidance are not fully detailed, do not provide all possible details regarding the data requirements, and more than that, the approaches to generating data, collecting data, appraising data, synthesizing data. So for these cases, the guidance can turn to be useful because it provides examples and instructions on how to plan these aspects.
Yes,
so,
but for the regulated products, I mean, there are the regulation itself sort of provides the structured framework for the assessment. And I think I will jump to the question that is sort of maybe a continuum of this one. Will the implementation of protocols become compulsory in all EFSA opinions, generic and regulated? And if so, when will this happen? And it's maybe good if we could clarify the generic versus regulated and the level of compulsory. If you could maybe continue, Elisa, answering that.
Yeah. According to our strategy and also quality management system that helps implement the strategy, protocols are now a requirement, so a mandatory requirement for EFSA generic mandates only. Not for assessments of processes of regulated products, because as we said, for them there is already the legislative framework. That does not mean that all EFSA protocols for generic mandates will have to be extensive and detailed and will have to be published for public consultation before being finalized, etc. Because, as this guidance explains, the approach to protocol development must be flexible, dynamic, and tailored to the specificities of the mandate. So this is, and the guidance helps implement this requirement set out in our quality management system.
Okay, thank you. We have a question. Who will define the protocol? EFSA staff in a given panel, a cross-cutting working group, and so on. So I think maybe Laura, if you could comment on this one.
Yes, thank you for this question. Actually, the protocol goes through the normal process that we have in EFSA. So generally speaking, we have working groups that work on the definition, working on the mandate. And actually this starts in the generic domain with the protocol development, and then the protocol is revised and endorsed first and then adopted by the panel to which the working group refers to, and can go through public consultation if this is agreed with the mandate requester during this negotiation phase that Elisa has already mentioned. But for the rest, there is no actually difference with what we do normally when we develop any assessment. So the difference is only at the level of the publication, whether it go public, whether it go for a targeted consultation, whether we have a workshop on it, but for the rest, it is very similar to what happens with the assessment process.
So basically, it is the group of experts or a subgroup of experts along with EFSA staff that are going to carry out the assessment to do the planning upfront. Yeah. We have a question. Does the guidance contain any example tip on how to apply it to the development, to develop a scientific guidance document on plant protection products, for example? Elisa, maybe?
Thank you for the question. The guidance does provide quite some examples, mainly on the problem formulation part and this a priori paradigm that will be presented after the coffee break. Not specifically on plant protection products because they fall into the case of regulated products. So they are not, let's say, the target of this guidance. There are also some practical examples on how to apply the guidance in the so-called template for protocols that is annexed to the guidance and is also now publicly available through the public consultation for information. This document complements the guidance and provides examples of how to apply the guidance.
But maybe as a follow-up in terms of the question, so
Dora, wait, sorry, you're not live. Okay.
Be a follow-up question from me on that question for clarification. So when this guidance on protocol development, I mean, what is the applicability of this guidance for mandates that are sort of directed at developing guidance documents within EFSA? Are they exempt from this protocol development, or sort of could you maybe clarify for the audience a little bit? Because that's how I understand question number 5 in sort of how the guidance can be applied in general terms to the development of scientific guidance documents.
It is not meant for developing scientific guidance documents, although the way I interpret this concept of guidance document, I might be wrong, is, for instance, for the field of plant protection products as a scientific assessment. So where data are collected, appraised, synthesized, conclusions are drawn. So it's a scientific assessment process. The guidance is not meant for these types of scientific assessments because those are assessments of processes of regulated products. Correct me if I'm wrong, if my interpretation is wrong. Maybe the person who posted the question can tell us if we properly addressed it. Thank you.
And I think also the questions that we've answered so far, if you have any follow-up questions or something wasn't clear, please pose a question in the chat and we will try to clarify. There is a question on problem formulation. It was mentioned that a priori is the recommended approach, but not compulsory. Can you tell more about when to apply it and examples on when other approaches might be more appropriate? And maybe Laura, if you could come in on that.
Sure. Thank you for the question again. We are going to discuss more in detail about the a priori framework in the presentation after the coffee break. But just to say that the reason why we recommend the a priori approach to anticipate that it is really because we felt the need to have a paradigm that could be broad enough to cover the breadth of the kind of question that we receive in EFSA. So it is not compulsory in the sense that there are others, for instance, the PICO-PECO, that could fit well certain domains. For instance, it is very well suited for the clinical trial setting. But as we will see in the next presentation, then there are also limitations in this approach when it comes to other domains, such as biological hazard, for instance, plant and animal health. But I suggest that the attendee waits a bit and then maybe poses a second question if he or she is not satisfied with the presentation after the coffee break. Thank you.
Thank you very much. We have another question. The mandate is broken down into problem. The mandate is broken down in the problem formulation to assessment questions and sub-questions. Especially sub-questions need to be operational. Does this include criteria on how to define acceptable harm or biological relevance of test results? And maybe if I give the floor, answer a part of that question first and give the floor to Elisa, I would think that there are existing guidance documents on biological relevance and how we sort of define harm. And I think that those guidance documents apply and are taken into consideration when we develop the sub-questions. But Elisa, if you would comment on this. But that would be the answer to the question. Yeah, that the existing guidance documents on biological relevance, we have those. I mean, we have standard procedures and how we define harm. Of course, they can vary between fields, so that would be the experts, and the sub-questions would not override that. We have another question. Is the protocol development phase included in the mandate timeline, and how long does the phase take normally? And I think this is a very good question, and maybe Laura, if you could comment on this.
Sorry, I couldn't unmute myself. Indeed, this is a very good point. And we know that the protocol development can take time, particularly the first time that is done. Then over time, what we have observed that there is a learning curve, and actually the time needed for this process can low down. But it is important to properly plan as part of the mandate also the protocol development. And since now, as said, it is mandatory and it is also foreseen as part of our scientific assessment process and the negotiation, it is in a way becoming a standard, so we would expect that over time, again, it will require less time.
And I think if I can follow up on that, sort of based on experience, I mean, the protocol some of the protocol during the implementation phase work was sort of high level, sort of large mandates that required a lot of time and effort to produce. But the guidance document that we have before us drawing on the experience is that we need to be flexible and be proportional to the mandate and the time frame set out. And I think people are getting more and more used to doing this protocol up front, and people have been generally quite happy about that. You know, the time that you use to develop the protocol will end up saving you effort further down the road, but it's an adaptive phase. But maybe Elisa, if you would come further in, do you have any more comments on that? No. Then I don't see any. I'm not live. I don't see any more questions in the chat at the moment, so we have still some time left. So if there's anything that hasn't been answered, please state them now before we go to the coffee break. And I will just wait maybe a little while. But sort of to sum But just to summarize, I mean, we had a question on whether we expect protocol will influence the way the European Commission gives mandate to EFSA, and the answer to that question is basically no. We think there will be interaction and we will have more clear mandates and sort of more clear picture on how to do the assessment in the future. I mean, the concept doesn't really influence the protection goals. I think there was a question on that. And the guidance may be applicable to sort of, it is primarily meant for generic mandates, but so that was one question, and in principle it is the sort of the working group experts along with EFSA staff that develop the protocol. We had a question on protocol being compulsory or not, and I think the aim is that we do protocols for all generic assessments in the future, and then the guidance is applicable to other areas. And I think the questions on APRIO will become more clear after the coffee break. So if there are no further questions, I suggest that we stick to the timeline and we start 30 past. Thank you very much for this session.
Welcome back, everybody. I hope you all rejoined after the coffee break. I would give maybe 1 more minute to people to reconnect for this second part of the info session that will focus on quite an important component of the problem formulation, that is the new a priori paradigm. And we will first give a general introduction and then move to 4 illustrative examples that will be given by 4 colleagues. So let's wait 1 minute more and then we restart. Thank you. Okay, it is 31, so let's start back with the details on the newly formulated a priori paradigm. And we have seen in the previous presentation that the outcome of the problem formulation is a list of operationalized assessment questions as questions, so questions that can be addressed from a scientific viewpoint. And we all know that this process can be quite challenging, and therefore the availability of a structure framework can be of help during this process. This is the reason why we believe that the a priori paradigm can be useful. And just to give a bit of history that was a bit anticipated by Thor, this paradigm was initially developed by our contractor, Risk Science International, that came out with this proposal after analyzing around 700 EFSA scientific assessments that were published in the years 19 and 20. And then it has been revised by the working group on protocol development and adopted and included in the guidance. But if you are curious to know more about the deliverable of the contract, you can have a look at the section 2.3 of the guidance. Next, please. So let's start with the first element of the a priori paradigm, that is the agent. That is a very broad term that includes any cause of an effect on a receptor. And you see in this slide some examples taken from the various EFSA domains that give you a sense of the real broadness of this term, since it can span from, I don't know, natural toxins, such as mycotoxins in the contaminant field, but it can be also nutrient in the nutritional field. But if you look at the animal health and welfare domain, it can be identified as a slaughter method. So there is really quite a variety of ways in which the agent can be characterized. Next, please. The P stands for pathway. That refers to any way in which an agent can interact with its receptor. And it can also refer to the source of an agent, for instance, the introduction to an environment. And again, you can see a few examples displayed here. The pathway can be a deliberate addition during feed manufacturing when it comes to additives that are used in feed. Could be transcutaneous transmission in the biological laser domain. Could be non-dietary pathways such as dermal inhalatory when it comes to contaminants. Just to mention a few, but you have a full list of the characterization of the pathway and the other elements of the a priori in the table 2 of the guidance. Next, please. The R stands for receptor, and it refers to anything that experiences the effect of an agent. And this experience can be also a sort of secondary consequence to the exposure to the agent. That could be the case of a farmer that changed the cropping practices as a consequence of the crops being affected by a pest. And you see again some examples from the specific domains. So the receptor can be the obvious human population of the consumers, but can be also genetically modified maize, could be ecosystem in the plant health, could be cell lines, organoids, when we deal with in vitro evidence for food additives and maybe also for other types of questions. Next, please. Next. I don't see the next slide. I don't know whether it is only me. Okay, so the I stands for intervention, and it refers to any intentional measure that is aimed at changing directly or indirectly the exposure or the consequence of the exposure to an agent. And I would like to spend a few words more on this element because this term has been traditionally used with a different meaning in other domains. For instance, in the well-known PICO framework. And in that framework, it refers to actually the effectiveness of a substance in clinical trials, for instance, the drugs. Here, as you can see, it has a quite different meaning in the sense that it is really a measure of which we want to measure the impact. So it could be, for instance, a biosecurity measure in the animal health and welfare, or could be the eradication in plant health, could be the pathogen growth inhibition measure for biological hazard. Next, please. So the last element of the a priori is the O, the output, that is really the form taken by the answer to the question or sub-question. And this form can vary a lot. Again, can be the pathogen load reduction with and without control measures in biological hazard, could be the description of the characteristics of the agent for the plant protection products, could be the dietary reference values if we look at the nutritional field. Next, please. And it is important to stress that the meaning and the characterization of the a priori elements really depends on the type of question attends. And I think it is useful to clarify how, for instance, a treatment can be characterized depending on the specific question in which it is addressed. So, for example, in an efficacy question, whose aim is to really assess the effect of different treatment conditions in an experimental setting, not in the real condition. So it could be, for instance, a sub-question that aims at assessing the efficacy in terms of log reduction of pathogen load of the high pressure processing, a different time pressure combination on a pathogen in milk in an experiment. And if you look at this kind of question, then the treatment is the agent, since it is the one causing an effect, so the reduction. The receptor is the pathogen in milk. And this can be generalized to other type of treatments, such as feed additives, and it impacts on the impact on the aflatoxin in feed. And in this case, the concept of pathway is not very relevant, whereas it is very important for the intervention question, in which you might have a similar treatment, but addressed in a different way, since you could have a question aimed at assessing the impact of the high pressure processing in the milk processing at a specific step of the food chain and at a specific combination of time and pressure on the probability of listeriosis in the elderly human population. So in this type of question, the pathway is fundamental, since it helps identifying how and when the intervention is applied to change the consequences, that is the probability of listeriosis, following the exposure to the agent, that in this case would be the listeria and on the receptor, that is the elderly population. So I hope this helped clarifying the difference between the two. And then moving to the advantages that we see also to reply to, in a way, to the question that we received before, why we felt the need to introduce a new paradigm. First of all, the APRIO framework offers a formal and structured approach for the formulation of the question and sub-question. It specify explicitly some components of the questions, such as the output, the O element, and the pathway that is missing in other paradigm. And this is very helpful when it comes to the identification of the evidence needs. It is very adaptable to the specificity of the assessment, in the sense that not all the questions and sub-questions requires the specification of all the APRIO elements. For instance, the I, the intervention, is only needed for the intervention questions, and the pathway generally is relevant for question in the plant and animal health and biological hazard domain. And it is also very broadly applicable within and across the various EFSA domains. So the expectation is that it will foster the harmonization and the consistency in the protocol development also across the EFSA scientific assessment. So to conclude, the APRIO provides a logical cross-cutting approach that is deemed useful in all types of EFSA scientific assessments. And I invite you to have a look at the section 6.2 of the guidance if you want to have more details on it. And examples of the APRIO implementations in various EFSA domains are provided in the guidance in the Appendix A, but we will give also some examples in the next presentation. And therefore, I give the floor to my colleagues, Kostas Koutsumanis and Winnie Messens, that will present a first example on the high pressure processing. Floor is yours.
Thank you very much, Laura. I don't know if you can hear me well.
Yes.
Okay, great. So the first APRIO example is actually related to high pressure processing of food. As I said, this is not a real example, but it is inspired by a previous opinion of the biohazard panel of EFSA. So in this slide, you can see some information about the high pressure processing of foods. So high hydrostatic pressure is a non-thermal actually treatment in which foods are subjected to isostatic pressure. And for most application, we have solid and liquid food which are processed in the package. And this is the main characteristic of this processing. It is mainly applied for the non-thermal inactivation of non-pathogenic, both pathogenic and spoilage vegetative microorganisms. And the objective is actually to increase the microbiological safety, but also to extend the shelf life of processed food. And high pressure can be applied at different points in the food processing and preservation chain, and it has in general a minimal impact on thermally sensitive compounds like nutrients and vitamins, and this is the main benefit this processing, and of course in sensory characteristic. If we go, so the two questions we have in this hypothetical mandate. The first is about the efficacy and microbiological safety of the high pressure when applied to relevant foodstuffs. And the second question, the TOR, is the efficacy of the high pressure when applied to raw milk and raw colostrum from ruminants. So Winnie Messens will proceed with more details about the a priori example on the high hydrostatic pressure. So please, Winnie, the floor is yours.
Thank you, Kostas. So as you can see in this slide, the first TOR, or terms of reference, was split into three sub-TORs. The first one, TOR 1A, asks about the foods to which high pressure processing, or HPP, is or could be applied along with the processing conditions. Then we can think about pressure intensity, duration of treatment. The second sub-TOR, TOR 1B, requests to list those factors that may influence the efficacy. And the efficacy, it's actually the reduction of pathogens using the high pressure processing. So those factors can be like the food itself or the processing related factors. And then the last TOR 1C asks about the microbiological food safety concerns of pressure treated foods. So this is in fact broader than the efficacy. As you can see, all the sub-TORs were translated into a single assessment question, and only the first assessment question 1 was broken down into two sub-questions. One requesting about the food categories being treated, while the other one asks about the high pressure processing conditions. So on this slide, you can see the implementation of the a priori framework, which is actually quite straightforward in this case. This is because all the assessment questions and sub-questions can only be kind of implemented in the a priori framework using the element output. So, or the type of answer that actually needs to be specified. And this is because the question is descriptive. As an example, we are only asked to list the food categories being treated in sub-question 1.1 or the processing conditions in sub-question 1.2. However, it is still important to consider well, so in the previous slide, to consider well what is actually being requested. For example, to clearly define that the focus is on commercial processing and that actually the foods are being treated with the aim to increase food safety. So in this case, actually the a priori sub-question can be directly derived from the formulated output. The next slide brings me to the term of reference 2, which actually is also split into three sub-TORs. The TOR 2A asks to derive minimum requirements of the high pressure processing for the treatment of milk specifically, and that in terms of pressure and duration, in order to achieve an equivalent efficacy to the traditional thermal pasteurization of milk. And this TOR is translated into the assessment question 4. Just as a reminder, so the efficacy was used here as the impact of those treatments on the reduction of pathogens, and which pathogens to consider for this are spelled out in sub-question 4.1. So again, here the request is a descriptive listing of the pathogens, and thus only in the a priori framework, only the output needs to be spelled out. On the other hand, for the efficacy or the inactivation of the listed pathogens, that is then evaluated for thermal pasteurization, so in one sub-question, and using HPP in the other sub-question. In the next slide, you can see how a priori is being implemented here. And you can see that actually, as Laura already explained, here we have the typical situation of an efficacy question. because the aim is to assess the effect of a treatment. So in this case, thermal pasteurization or HPP. The treatment, it's now the agent or the cause of an effect, while the receptor or what experiences the effect of the agent or of the treatment are the pathogens of concern and that are present in the raw milk. The output is the achieved log reduction of those pathogens as achieved through the treatments. So then bringing all these a priori elements together helps to formulate the sub-questions. The next slide then gives the assessment question 4, which it's now actually answering this TOR 2A. And this is actually using the reduction of the pathogens obtained through the thermal pasteurization and provides those time pressure combinations using HPP that give at least the same reductions or so-called minimum requirements. So now the output has been changed to reflect this. Then in the next slide, it is presented the term of reference to C. And in this case, it asks to compare the risks to public health by consumption of 4 types of milk: pressure-treated, untreated, or raw milk in that case, versus pasteurized milk and ultra-high temperature treated milk. And actually for these latter 2, using the legal requirements. This TOR2C was translated into an assessment question that compares the pathogen load in the 4 types of a milk serving. And in the a priori field, we are in the situation of an intervention question. The intervention or the measure to change the exposure to the agent is the treatment introduced in real-life situations in comparison to no treatment or other treatments. And in this case, the contaminated milk batch is the receptor of the treatment, while the relevant pathogens are the agents. The output is the pathogen load and the probability of contamination in a milk serving. The next slide gives an overview of the conceptual model. It shows how the different assessment questions and sub-questions are related. Also, within parts of different term of references. So as you can see using the different arrows. For example, in orange, you can see that the high pressure processing conditions are used in various questions. So then at last, allow me to say that the Appendix A4 also contains the example of a classical QMRA, quantitative microbial risk assessment, containing exposure assessment, hazard characterization, and risk characterization. So now I give the floor to Susanne Bennekou from the Danish Patient Safety Authority.
Many thanks, Winnie. And before I present this case here, I just want to remind the audience that please post your questions in the Q&A for the discussion we will have after these examples. So I would like to present a case on aflatoxin B1 in food. And so here we start with an example where we would like to illustrate how the a priori paradigm can be used in a quite a complex mandate that would encompass several EFSA domains and as such, just to show how it can be applied and be useful. Now, please stay here. Yeah. No, the previous slide. Yeah. So we start out with the biological pathway that starts with the fungus Aspergillus that would impact, for example, nuts and seeds, crops, and it can proliferate there, and depending on specific climate conditions, it could lead to increased concentrations of mycotoxins. And of course, So these mycotoxins could affect human health via dietary exposure, contaminant in the diet, and they can also affect animal health as a contaminant in feed. Furthermore, you can reduce the bioavailability of the mycotoxins if you add certain feed additives like bentonite, which binds or capture aflatoxin B1 in feed, and thus you would have a reduced exposure, and this would again carry over to a reduced content in milk, for example. So this is an example where we have actually a biological pathway that encompasses three major areas of EFSA. And here we try to illustrate that in the conceptual model we have here. So we have the mandate 1 with relating to the plant health. We have mandate 2 relating to contaminants, both in food and feed. And then we have the mandate 3 as feed additives. We have here again tried to capture how how the a priori elements are different here in the different examples in the different mandates here. So for all cases, the agent would be in case 1, mandate 1, the agent is the Aspergillus fungus, while in mandate 2, it's the aflatoxin, mycotoxin, and then the agent in mandate 3, for example, is the feed additive. And then again, we have different pathways here, depending on the mandates, different crops, different receptors. We have in mandate 1, in this case, it's the plant health, the plant. For the contamination, contaminants, it's the European population or farm animals. And then in case of feed additives, the receptor in this case would be the aflatoxin, mycotoxin. Here we also, in related to plant health, we can apply practices that would reduce the content and or the viability of the fungus and thus the production of the mycotoxins by applying different cropping practices, while for the other 2 mandates we don't have any intervention. And for all, of course, for the outputs, these are again different in these cases. For the plant health, it would be the probability of having the Aspergillus infection related to cropping practices, and then related to contaminants in food and feed, it's adverse effects. And then related to feed additives, it's a decreased level of the mycotoxin in the feed. And this has then been translated into the assessment questions. And I think the good thing about also illustrating the conceptual model here is that you can really, by these arrows, indicate how or illustrate the whole biological pathway and how the mandates relate to each other. And I think this shows the versatility of the a priori questions, because if it's harmonized, the way you phrase these questions and how you develop the protocols, there's a good opportunity, or that you should be able to read across between the mandates. And this is actually what we really would like to illustrate here with this example here, that with the use of a harmonized approach here, it would be easier to integrate different elements from different protocols in different contexts. Now I would like to give the floor to my colleague Louisa. She will flesh out a bit more on the sub-questions for one of the terms of references we have presented here. So please, Louisa.
Thank you very much, Susanne. So to exemplify the use of the a priori paradigm in the field of contaminants, and if we can go one slide back, please. I will focus on a hypothetical mandate related to the presence of aflatoxin B1 in food, as you see it highlighted in the slide. Next, please. The genetic formulation of the assessment question for this hypothetical mandate could be, what is the risk for human health related to the presence of aflatoxin B1 in food? Next, please. Using the a priori, we can refine further this assessment question by defining the agent, aflatoxin B1 in this case, the pathway, chronic dietary exposure in this example. The receptor would be the European population, all age groups, intervention not applicable, as has been explained before, and the output would be the risk of adverse effects. So this would result in the a priori formulated assessment question, what is the risk for human health associated to chronic dietary exposure to aflatoxin B1 in the European population? Next, please. The risk assessment of chemical contaminants follows the 4 steps that are highlighted here, also known as the 4 pillars of the risk assessment. So the hazard identification and hazard characterization that has the final aim, if possible, to establish a health-based guidance value, for example, a TDI or a TWI. The exposure assessment, and then the final steps, that is the risk characterization that combines the previous ones, so relates the exposure to a chemical in a given population with toxicological effects and concludes on the likelihood of adverse effects. Next. If we go back to our hypothetical mandate, we can base the formulation of the sub-questions for each step that we have seen using the a priori paradigm. And this is what is illustrated in this slide and is also shown in the appendix of the draft guidance. Next, please. Focusing on one of these sub-questions, the one related to the exposure assessment, and if we use the a priori paradigm, in this case, the agent would be still aflatoxin B1, the pathway would be the chronic dietary ingestion, the receptor would be the European population, all age groups and countries, intervention not applicable, and the output would be the mean and P95 estimates of the chronic dietary exposure per kilo body weight per day. So you see on the right side of the slide how it would read the a priori formulated sub-question. What is the estimate of the mean and P95 chronic dietary exposure per kilo body weight per day to aflatoxin B1 in the European population? Next, please. From these further sub-questions can be and should be further formulated to inform and to be able to reply to this sub-question related to the exposure assessment. And this sub-question should relate to the occurrence in food, to the food consumption data, and, for example, also to the food processing. Next, please. In this slide, we see how a priori can help in formulating the sub-question related to the occurrence in food. Agent aflatoxin B1, the pathway would be contamination along the food chain. The receptors would be the food items codified according to Food X2. And the output would be the mean occurrence in each food item. And on the right side of the screen, you can see how the a priori formulated sub-question would read. Moving now to the food consumption. Next slide, thank you. In this case, the agent would be the food items potentially contaminated with aflatoxin B1. Pathway, we are still interested in the chronic dietary ingestion, receptor, the European population, all age groups, and the output in this case would be the individual chronic consumption data adjusted by body weight from the surveys included in the EFSA comprehensive database. And having this information will help us to formulate a more rich and clear sub-question, as you see in the right side of the slide. Next, please. The last example of a sub-question related to the exposure assessment could be related to the food processing. And in this case, different from the others, the agent would be the industrial and household food processing, milling, heat treatment, boiling, etc. The pathway would not be applicable in this case. The receptor would be the food items potentially contaminated by aflatoxin B1, and the output would be the percentage of reduction or increase in the occurrence after this processing or storage for each food item. And the complete a priori formulated sub-question, it is shown in the right side of the screen. Before finishing this example, I would just like to highlight that this is an hypothetical mandate to exemplify how a priori could help in the problem formulation, and that other sub-questions are possible besides those presented here. And with this, I would like to give the floor to my colleague, Andrea Gerbel-Meyer.
Yeah, thank you. I'm now going to present to you a hypothetical assessment in the area of EFSA's panel on animal health and welfare involving a pathway to harm. Next, please. The assessment concerns a virus that many of you will have heard of due to the ongoing outbreaks in many European member states. Highly pathogenic avian influenza virus, in short, HPAI. The hypothetical term of reference that EFSA could receive from the European Commission is shown here. What is the impact of possible control measures on the probability of introduction of highly pathogenic avian influenza H5N8 into EU poultry holdings via wild birds? Next, please. Now, before we dive into the problem formulation using the a priori approach, I would like to give you a bit of background on the disease and its epidemiology. So the HPAI virus usually reaches Europe via infected migratory birds, wild birds. When they arrive in the EU, they rest near lakes and feed in fields, which they then contaminate with infectious virus that they shed with their excretions. The European wild birds that are also using these habitats can then become infected with HPAI while they are feeding or swimming or whatever they do in these areas. And these infected EU wild birds are excreting the virus in areas where we have poultry farms. Now, let's have a closer look at how infected wild birds can infect domestic poultry with the virus. Next slide, please. So the obvious introduction pathway is contact between infectious wild birds and poultry that is kept outdoors. The infected wild birds could become into direct contact with the chickens or contaminate their feeders with the virus. So that's why poultry is kept indoors as soon as HPAI has been identified in the area. But HPAI can also be introduced indirectly, that is through boots, clothing, farm equipment, and many other items that have been in contact with infectious virus in the environment of the farms. So, for example, if the farmer walks through excretions of infected wild birds in the fields near his farm and then enters the poultry house with the same boots without cleaning them, he carries the virus directly to his chickens, thus seeding a new outbreak of HPAI among his flock. Next slide, please. So after this brief outlook at the disease and its epidemiology, let's start to analyze the term of reference. The agent is highly pathogenic avian influenza virus H5N8. That's a special subtype of the virus. The pathway is contact with excretions of infected wild birds. The receptor is poultry flocks in the EU, and the intervention are control measures that are not further specified in the term of reference. That could be washing your boots before entering the stable or wearing disposable boots. And then the output that we are interested in is a bit complicated because it's the impact of possible control measures. So impact means that we have to make a comparison, and that would be the ratio of probability of virus introduction with control measures as opposed to the probability of virus introduction at baseline, so without control measures. Next slide, please. So based on this, we can formulate the assessment question like this. What is the probability of introduction of HPAI, H5N8 in poultry flocks present in EU holdings applying control measures compared to baseline probability. But obviously, if we think of what I've earlier explained about the epidemiology of HPAI, there are a couple of sub-questions into which this assessment question has to be broken down. Next slide, please. So the first concerns the introduction of HPAI through migratory wild birds into the European wild bird population. Next one. For this step of the pathway, we have to consider two issues. The first is the prevalence of the virus among the migratory wild birds entering the EU. And for this sub-question, the agent is obviously HPAI H5N8. Then the receptor would be the migrating wild bird species that are entering the EU. And the output would be the average prevalence in a typical year. That means the number of infected birds among all of the birds. So our sub-question would be, what is the average prevalence of the virus in migrating wild birds entering the EU? All species in a typical year. So the other issue we need to assess is the number of migratory birds entering the EU. Here, regarding the a priori elements, we are concerned with the receptor, which would be the migrating wild birds entering, and the output, the average number across all species in a typical year. So as you can see, analyzing the terms of reference in this way not only helps to understand the scope of the assessment needed, but it also highlights the data that is needed as well as the expertise. In this example, it's evident that we need to involve people with expertise regarding a range of wild bird species, such as ornithologists. And we would also need data about wild birds, which is not available from veterinary services. Next slide, please. So the second sub-question concerns the spread of the virus among the wild birds present in the EU. Next slide, please. So again, we could look at two sub-questions here. The first being the size of the population of EU wild birds, and the a priori for this one is very simple. It concerns basically only the output. What is the size of the EU residential wild bird population? The second question regards the probability of contact of EU residential wild birds with infectious excretions containing viable virus. And here, in the a priori would have as the agent the infectious excretions containing the virus, the receptor, again, the EU residential wild birds, and then the output would be the probability of contact with the infectious excretions. And based on that, our question would be, what is the probability of contact of the EU residential birds with the excretions contaminated with the virus? And if you look at the second sub-question, one immediately starts to think about how would you know that? So here, this approach of problem formulation using the a priori also assists in identifying useful methodological approaches. And the one that comes to mind here is expert knowledge elicitation. Okay. Now, let's look at the final step of our hypothetical assessment, the introduction of the virus into EU poultry holdings. As said at the beginning, we need to compare two scenarios. The upper one being the introduction of HPAI to poultry holdings when no control measures are applied, and the second one being the introduction of HPAI to poultry holdings when control measures are applied, such as wearing disposable boots. Next slide, please. So again, we could use two sub-questions for this step. For the baseline probability, we would have all elements of the a priori except for the intervention. And the sub-question would be, what is the probability of introduction of H5N8 into EU poultry holdings through contact with wild birds' infectious excretions introduced into the poultry holdings in the absence of control measures? And for the second sub-question, everything
would be the same except for the intervention that we would have here in the form of, for example, washing boots before entering the premises. Next slide, please. So, in summary, I hope I have been able to demonstrate that the a priori approach can also be used for pathway to harm assessments, and that it is very helpful for teasing out and formulating the different assessment questions and sub-questions included in the TORs that we receive from our requesters. In addition, I hope I've succeeded in illustrating that the a priori approach can also assist in identifying the data that we need for our assessment, the experts that we have to bring into our working groups, and the methodological approaches that are useful in addressing these questions. So with this, I end my presentation and I hand over to Andrew Kraft for another hypothetical a priori example. Thank you.
Thank you, Andrea. So I'm going to present the last example of the a priori. It's a hypothetical non-cancer hazard assessment of formaldehyde in air. Next slide, please. So first, my disclaimers. So I'm an employee of the US EPA, and I'm presenting my views and not necessarily those of my agency. And second, this hypothetical a priori example is modeled after a publicly available draft EPA assessment. However, the two are unrelated. Next slide, please. So the generic question we're asking here is pretty straightforward. It's maybe a little more simple than some of the other examples you've heard today, but it's what is the level of lifetime inhalation exposure to formaldehyde expected to be without adverse non-cancer health effects in the general population. So you'll notice there's some overlap with the prior example on aflatoxin, as well as some of the points that Laura covered in the session earlier today are going to be kind of covered here as well. Next slide, please. So as some background, in case you're not aware, formaldehyde is a very well-studied high production volume chemical. It's pretty much ubiquitous in both indoor and outdoor air in the United States. Because it's very well studied, there's a number of assessments on this chemical and its potential health effects. And this knowledge was used to inform the framing of the a priori assessment question, or AQ, and the sub-questions. In addition, as you'll see in this example here, a large level of detail in terms of higher level tiers are presented so that this might better facilitate the application of principles of systematic review during assessment development. I think we've already covered what hazard assessment is, so I'll skip that. Next slide, please. So as a preliminary step to developing the conceptual model, although persons can be potentially exposed to formaldehyde from the oral, inhalation, or dermal routes, this example will focus on inhalation exposure alone. As far as receptors go, we're interested in any populations or life stages, including potentially susceptible individuals that might be exposed to formaldehyde via inhalation. And here we're going to use the existing knowledge to frame the a priori around specific non-cancer health effects that might be affected by formaldehyde inhalation exposures. This is previously studied information on formaldehyde inhalation, so focusing specifically on the non-cancer health effects of sensory irritation, pulmonary function, respiratory tract pathology, asthma or allergic conditions, nervous system effects, male reproductive effects, female reproductive effects, or developmental toxicity, and all other health effects are kind of outside of scope of both this example as well as the assessment. And then in terms of dose response analysis, that will be applied only to those health effects interpreted with sufficient certainty of the evidence for a hazard, as I'll get to later. So using the a priori paradigm to formulate the assessment question, we get a little more specificity on that generic question. Here it's what is the airborne formaldehyde concentration below which adverse non-cancer health effects are not expected to occur in any person, including susceptible individuals exposed for a lifetime. So you can see this is broken out on the right with the a priori, the agent here is formaldehyde, the pathway is lifetime inhalation exposure, the receptor is the general population, including potentially susceptible individuals. Intervention is not relevant to the assessment question or to any of the sub-questions I'm going to present. And the output here is that airborne formaldehyde concentration below which effects are not expected to occur. Next slide, please. So that assessment question is broken out into two tier 1 sub-questions. Can you go to the previous slide, please? The first sub-question is on hazard identification for those non-cancer health effects. And the second one will be on the dose response analysis, which I'll present later. And you'll see this is consistent with the other examples that were presented by Laura earlier, as well as for aflatoxin. So for this tier 1 sub-question on hazard ID, we're asking generally, what are the specific non-cancer health effects expected to result from inhalation exposure to formaldehyde for a lifetime? And here when we say expected, we're inferring an expert judgment mediated decision about having sufficient certainty that the available evidence supports a hazard. So that'll become important later. And here the a priori framing that, the A, P, R, and I are all the same as that general assessment question, and here the output is that hazard ID determination. Next slide, please. So here that tier 1 sub-question on hazard ID is broken out into a number of tier 2 sub-questions for each of the various non-cancer health effects that are being examined. So this would facilitate a specific systematic review of each health effect. So you'll see here an example shown for sensory irritation, but you would also have a sub-question at this tier for pulmonary function, for respiratory tract pathology, etc. The general question being asked here is, does the health effect of interest, in this example, increased sensory irritation, represent a potential adverse health effect of lifetime exposure to formaldehyde via inhalation? And so you'll see the A, P, R, and I are again the same. And here the output is a hazard ID determination specifically for sensory irritation. Next slide, please. So for each of those tier 2 sub-questions on the hazard ID for the various non-cancer health effects, we would then have a number of tier 3 sub-questions that would need to be formulated using the a priori. The example shown here is a tier 3 sub-question on sensory irritation specifically related to the available human studies. You would also have a tier 3 sub-question for the available animal studies on sensory irritation, a tier 3 sub-question on the available mechanistic studies on sensory irritation, and potentially other tier 3 questions. And then you might also have tier 4 sub-questions that would inform the determination, for example, of the mechanistic data that might have multiple different models that need to be independently considered. So for the human data, we're asking, do the available human studies, and we have both epidemiological and controlled exposure studies available for this, support the determination of sensory irritation as a possible adverse health effect of lifetime inhalation exposure? Using the a priori again, the agent is still formaldehyde. The pathway is a little different here, because in these studies, the inhalation exposure may be a measured and reported formaldehyde air concentration. It may be a concentration that could be estimated, for example, through a job exposure matrix, or it can be otherwise inferred. Similarly, the receptor is more definitively defined here, because we're asking specifically about exposed humans in these studies, particularly in worker, residential, or controlled exposure studies. Again, the intervention is not relevant, and the output would be that hazard ID determination related to sensory irritation based only on the human studies. Next slide, please. So then turning to the other tier 1 sub-question for the assessment question here, this is on the dose response analysis. So during problem formulation, although one would develop an a priori for all of the non-cancer health effects that I mentioned earlier, during implementation, only a subset of those would potentially be answered by the assessment. So in this hypothetical example, perhaps the expert judgment resulted in a determination that only sensory irritation, pulmonary function, and respiratory tract pathology had sufficient evidence to support a hazard ID determination. The assessment would then only answer those sub-questions relevant to those health effects and not the others. So here we're asking can you go back, please? So here we're asking, what is the reference concentration, or RFC, for lifetime inhalation exposure to formaldehyde, below which adverse human health effects are not expected to occur in any person? And then what is the certainty of that RFC? And so we'll come back to that. So again, the A, P, R, and I are the same, and the output here is the reference concentration. Next slide, please. To answer this question, we would then have tier 2 sub-questions on each non-cancer health effect. I'm only going to show examples of those for which there was a determination of sufficient evidence. So sensory irritation is the example here. We would also do this for pulmonary function and respiratory tract pathology. And for each of these, you would want to determine a toxicity value for lifetime inhalation exposure for that particular health effect. So this is showing the example of sensory irritation, as well as the certainty in that value. And that certainty becomes important because this determines, one, whether we can even derive a toxicity value for that health effect with sufficient certainty, as well as how that toxicity value for each health effect feeds into that overall RFC determination. So here we again have the A, P, R, and I the same as before, and the output is a toxicity value specific to each health effect at this sub-question level. Next slide, please. I'm not going to go through this one in any detail, but basically for each toxicity value for each health effect, You would then have candidate toxicity values that you would need to derive and have a level of certainty in to inform that toxicity value for each health effect. So that's what's shown here. Next slide, please. And then to get to those candidate toxicity values, we would have to have points of departure from the individual studies or multiple studies. And here we're asking a tier 4 level sub-question on dose response. What are the best supported and most appropriate points of departure, or PODs, across the studies based on a list of ideally pre-specified criteria, given the available evidence on each health effect, in this case, sensory irritation. So here, formulating that using the a priori paradigm, we still have formaldehyde as the agent. The pathway would vary based on each study. So this is the study-specific exposures used to derive the POD. The receptor, again, would be study-specific populations or models that could be used to derive the POD. Intervention still not relevant, and the output would be those PODs as well as the relevant uncertainties. And this is my last slide. I think that's the end of the examples in the APRIO, so I'll hand the floor back to Tor.
Thank you very much. And I think it would be reasonable to explain that, the last example, which I like very much, is a good example of, it's non-dietary exposure, and somebody might wonder, why are we dealing with having that type of an example here when EFSA deals with dietary exposures? But the fact is that we are being asked more and more to take into consideration non-dietary exposures in our assessments. And I think the last example pretty nicely highlights the flexibility of the APRIO. Now, we currently have one question on relating to this section, and I would encourage you online to ask more questions if you have on the presentations, but the question that we have so far is directed to Louisa for slide number 74. Food processing is an intervention, is it not? So why is intervention not applicable here? There are different interventions that are being looked at.
If I can take this question,
yes, Laura,
as we agreed with Luisa. So maybe we can go back to the slide 74, but just to give a general explanation, we need to go back to the essence of the concept of the intervention, that it is really a measure that is deliberately introduced with a purpose to change either the exposure or the consequences of the exposure to the agent. So it really implies a sort of real-life condition, and also the fact that you interfere with the pathway between the agent and the receptor. And in the specific sub-questions discussed by Luisa, actually she was dealing with the efficacy question, in which you can think of a sort of experimental academic situation in which you are simply testing in an experimental setting what would happen potentially if you apply a treatment, food processing, to the agent, to the food that is the object of this processing. In this case, it was industrial and household food processing, for instance, take e-treatment, and the receptor is food item potentially contaminated by aflatoxin. And what you would do in this situation, you would simply try, for instance, different level of the temperature and the time to see how much the contamination of aflatoxin is reduced, but there will be any exposure at all, because you are in a situation that is not reflecting real life condition. It would be different if you take the same treatment, the treatments, and you look at the food chain processing at a specific step, and you want to see how, for instance, applying a specific temperature for a specific time in a specific step of the food processing would lead to a different risk of adverse effect in humans. So you are really interfering in the pathway. and with the exposure or the consequences of the exposure to the contamination in this case. And I hope this helps clarifying, but please post your feedback question if this is not the case.
Okay, we have another question. What happens if or when you misplace or mix up the aprio by mistake or lack evidence? For example, the A is assigned as R and vice versa. I think if I just start maybe by answering that, and before giving floor back to Laura, I think we discussed this quite a lot during the implementation and when making the examples, and the overall take-home message is I don't think there is anything as a mistake or a mix-up. It's a paradigm that you use to formulate your problems, and you can define the different elements in different ways to some extent. So there is no consequences there. But maybe Laura, if you would like to come back to this, sort of address this question briefly.
Sure. Indeed, as you said, there is also a level of flexibility in the implementation of the aprio, and the purpose of this paradigm is really to help and facilitate in the problem formulation process. So at the end, if it is more intuitive for someone to use this aprio element in a slightly different way, it is still acceptable. And what we say in the guidance is also that it is not mandatory to apply this paradigm, but we expect it to be actually helpful also because it will potentially foster also the harmonization, as we have seen, for example, in the nice examples that was presented by Suzanne and Luisa. There you have seen that possibly the same mandate could affect different panels, different domains in EFSA, and using this common paradigm would help to maintain consistency and harmonization across.
Yeah, and I think also with the aprio paradigm, I mean, when we compare sort of similar frameworks like PICO-PECO, they are defined around a very narrow area of randomized controlled trials in clinical medicine. But here we are dealing with anything from sort of chemical risk assessment, slaughtering of animals, inhalation, and so on. And there is I think we've gone quite a long way in terms of showing that it's relatively straightforward to formulate different mandates using this paradigm, but there is some sort of way to go when sort of it takes a little bit of time for everyone to feel comfortable, and therefore we stress in the guidance itself that we recommend this use, but it's not mandatory at the moment. But I think it will have a life on its own, and I think the experience has been quite positive. There's another question here. The same problem can be formulated in several ways, and that's absolutely true. Elisa, would you like to comment on this?
Okay, I can answer the
same.
Indeed, again, there is a level of flexibility in the sense that the purpose is really to have a tool that helps in this process that can be quite challenging, formulating accurately the assessment question and sub-question, because we know well that a well-formulated question simplifies a lot the life then when it comes to the identification of the evidence needs and also the definition of the methods to answer to this question. So that's why the intention was to provide a structured framework that though can be applied with a level of flexibility, and it really depends as a level of subjectivity, of course, the way the questions are defined. Also depends up to a certain level to the availability of the evidence, also on the resource available. So the question can be formulated in a broad sense, so can be then lead to a lot of effort, or can be narrowed down depending on the needs. So therefore, yes, there is room for ample room for differences in the way a question is formulated.
Yeah, maybe, I mean, it's the structure and the sort of that you are sort of, you're not, I wouldn't say forced, but you are asked to formulate a clear questions that fits into a certain framework. And even though you can do it differently also by convention, and we come back to that later in terms of sort of learning from each other and collecting examples of as we go along over time, I think there will be a sort of level of consistency will increase, and this will not be a major issue. I think now we still have a little bit room for further questions if anyone would like to ask. And I'll maybe give it a one and a half more minute if some questions drop in before we move on, otherwise to the next part. Okay, I don't see anything. Yes, I think it would be maybe just relevant, and we move on to the next section. And of course, you can still place questions on this session in the chat, and we will answer it by the end. But now I would like to give the floor to Fulvio Barizzone, who will speak about the methods for protocol development. Thank you.
Okay, thank you very much, Thor, and good afternoon, everybody. So next slides, please. Okay, here we are. So we are in the second step of protocol development. In the first one, we saw what we have to assess through proper formulation. And now we try to see how to assess those things. So namely, we need to specify the evidence needs and the methods for answering individual questions, then for synthesizing integration, then, and also to deal with all the surrounding uncertainty. Next slides, please. So let's start having a look at the approaches that we have for answering individual sub-questions. Basically, in EFSA, we have 5 main approaches to answer sub-questions that are, namely, using evidence from scientific literature or data from non-bibliographic databases. We can also even use expert judgment or mathematical or statistical models, or can also generate new empirical evidence. In any case, this approach can be conducted with different rigors of effectiveness. So think about, for instance, using scientific literature, that you can produce a narrative description of the literature, or you can have a systematic review, where, for instance, you arrive to a meta-analysis. Often those methods are used together in an assessment because they answer to different questions, in particular when we have broad mandates, and can also be nested the protocols can be nested. For instance, we may have a systematic review protocol nested in a more general protocol that try to depict the approach for answering a broad mandate. On the other hand, it can also be combined to answer a single sub-question. We may use evidence from scientific literature plus evidence that are coming from a non-bibliographic database to answer the same sub-question. In any case, there are always some common best practice to follow. So it's better if we have a clear question formulation of the problem at hand. If we use a multidisciplinary expertise, and we document the whole approach. Having said so, there is uncertainty that is inherent in all this process. That is due mainly to limitation in the evidence that we use and the approach that we apply. So this uncertainty should be characterized and documented through all the process, and all the sources of uncertainty should be minimized at every step of the approach, and if it is feasible, their combining impact of the final output should be assessed. Next slide, please. So let's now have a look at the first approach that we have to, let's say, answer individual question. So we're speaking about using evidence from scientifically literature study reports. We are speaking basically to using primary research studies that are available in bibliographic databases. And this evidence is frequently retrieved by EFSA via literature search. Nevertheless, sometimes can be submitted to EFSA by stakeholders, for instance, including an application dossier or submitted via call for data. In the majority of cases, this evidence comes in the form of aggregated information. We can think about the mean and the associated standard deviation, for instance, for a study group. And when we use this approach to answer the individual question, the process we follow is to formulate the question that we have, to retrieve the evidence. So that means that we need to define the data needs, the sources of information, and the search strategy that we want to follow. Then we need to select the studies for the inclusion. So again, we need to define the eligibility criteria. We need to extract the data from the inclusive studies. So we have a clear idea of the data model that we want. Then we need to appraise the evidence. And for this, usually we employ critical appraisal tool for doing that. And then we need to synthesize and integrate all this evidence accounting for the uncertainty. And finally, we need to analyze whether we have additional sources of uncertainty which were not previously addressed, and to express our overall certainty in the conclusion. Next slide, please. Another approach that we can follow is to use data from non-bibliographic databases. In this case, we are speaking about using evidence coming from searchable databases which have the information organized according to a predefined structure and that are accompanied by metadata. We can think about databases such as the EFSA Food Consumption Database or Eurostat, for instance. In this case, data can be individual or aggregated. A lot of time also we have individual data, and also can be represented by, for instance, the whole European level. That is the case for of Eurostat, for instance. Also in this case, the information are frequently retrieved by EFSA, but also sometimes are provided directly via dossier or a call for data. This can be ad hoc, or it can be a continuous call for data. We can think, for instance, the EFSA call for collection of chemical contaminant occurrence in food and feed, or directly in call for data that are prescribed by legislation. Zoonoses, antimicrobial resistant, foodborne outbreaks. And when we follow this approach, the main steps are represented by formulating the questions we have, collecting the data, so we need to define the data needs in the database or the provider, check and validate the information we have, and then also synthesize, integrate the evidence according for uncertainty, and analyze any further source of uncertainty. Next slide, thanks. Also, another approach could be the one of using directly expert judgment. And the expert judgment can be informally collected, for instance, via a discussion in an EFSA working group, for instance, or more formally elicited via what we call a semi-formal or formal expert knowledge elicitation, also EK. This form of expert judgment is mainly implemented when the value or the quantity of interest is not directly available by primary studies. And in this case, the elicitation process aims at trying to provide a reliable estimate for the quantity of interest and also a quantification of the uncertainty of in the estimate that we are going to make. It can also be used in proper formulation, for instance, for prioritizing the questions or in synthesis and evidence integration. One of the strengths of FACET is that it's allowed making use of the existing evidence at the same time reducing as much as possible heuristic biases and the influence of group thinking in a way which is transparent, reviewable, and also repeatable. Although we cannot be sure that to obtain the same result every time, because of course we are eliciting the judgment of the experts. So in that case, when we follow the EFSA guidance on ICE, for semi-formal or semi-formal level, the process defined there is the one of formulating the problem. Then we have a pre-elicitation phase in which we frame the ECHE question. Then we need to select the method and the expert for the elicitation and provide an evidence dossier. That is followed by the elicitation phase and a last phase of post-elicitation in which we are basically documenting the whole process. Next slide, please. Also, in some case, when, in particular for some EFSA assessments that are required remaining consideration in relation to the pathway to harm or pathway to firm or pathway to benefit, it's the use of mathematical or statistical model may be very helpful to address individual sub-question or the entire mandate or the assessment question. Of course, the use of those tools is conditional to the availability of input data that are of the appropriate validity. And when we use those, when we use models, basically already at the step of the proper formulation, the consideration starts with the criteria that we use to select or set the model that we have to use. And all other aspects of the assessment are set around the structure and the type of model that we need to use. This approach is very useful for evaluating the impact of some influential variables, for instance, or alternative condition, or maybe explain a phenomenon. And given the fact that it can predict things that are not already happening in, let's say, in real life, it's very useful for estimating the impact of possible intervention measures before their implementation. So it could be very useful for this manager. And when we use mathematical or statistical model, usually we follow the following steps. So we define the conceptual model, we identify the mathematical model we need to use, and then we identify the evidence needs for the parameter or variable quantification. Then we describe and implement the model. We verify and calibrate it. In case the model is newly developed or adapted, we need to validate it. And then we perform uncertainty and sensitivity analysis. And another approach could be the one or answering directly some question by generating new data via primary data collection. And this can be done by, let's say, collecting observation, setting up questionnaires or experiments. In those situations, using protocols is considered basically a common practice. However, planning of the methods can be very difficult and may vary depending on the type of study. In general, what happens at EFSA is that when we do primary research studies, these are outsourced to third parties and are done according to a pre-specified study protocol. Next slide. Okay, now try to summarize a little bit the situation. So what we can see is that all these approaches to answering individual question follow some kind of related stepwise processes, which all start with a clear question formulation and with some basically the same logic, I would say. They can be conducted with a spectral level of rigor and extensiveness. They can be used together, nested, or combined. And what is important is that we need, along all the process, when using them, we need to minimize the sources of uncertainty in each of those approaches. And when this is feasible, assess their combining impact on the outcome. Next slide. And then the second part of method specification is related to the specification of the methods for synthesizing and integrating the evidence. So when we speak about evidence synthesis, we are meaning about summarizing the evidence for a single sub-question at its finest degree of granularity. We can think, for instance, about let's say, putting together the results that are coming from different randomized control trials on the same topic. And in this case, the methods can range from qualitative, so we can have a narrative description of the result of all these studies, up to quantitative, where we can apply, for instance, a meta-analysis. On the other hand, when we speak about evidence integration, we are thinking about combining evidence from different study designs or line of evidence. So that would entail a higher level of heterogeneity overall and uncertainty. So, and then in any case, when we are speaking about evidence integration, we can have some basic integration scenarios. So we can integrate evidence within a sub-question, for instance, combining evidence from different study designs, across sub-question, so we can aggregate evidence from sub-question on different species to derive, for instance, a conclusion for humans, or across the assessment question. And of course, that depends a lot on the mandate sets of reference. In any case, in all these situations, a well-described conceptual model is key to define the process for evidence integration. Next slide. On the other hand, integrating evidence brings some additional challenges. For instance, when we try to integrate evidence from different study designs, we need to take into account the fact that there are uncertainties related to, for instance, the different abilities of those designs to represent real life condition or control for confounding. If we think about the experimental studies and observational studies, usually in experimental studies, confounding is controlled quite well. On the other hand, their ability to represent real life condition may not be the same one that we have in observational studies. Also, when we integrate evidence across sub-question, in certain cases, this could be a relatively straightforward process. So if we think about having an exposure assessment, putting together information about occurrence and consumption is relatively simple. However, sometimes this process can be very challenging. For instance, when we extrapolate the conclusion from in vitro strata to a target human population, that will be very, very difficult. So all these challenges translate into a set that must be then taken into account and, if possible, quantified. And it is fully recognized that planning the methods for synthesis and integration upfront can be very challenging, in particular when we don't have information about the possible data available, or there is a lot of degree of the heterogeneity in the information that we have at hand. A possible approach to, let's say, tackle this issue could be the one of setting up a stepwise approach where we set up alternative methods that at certain points that we can call decision moments can be selected on the basis of the information that are available at that point. So we may become familiar at a certain point with information that we have, so we can select a particular approach. Next slide, please. And then a last point related to uncertainty analysis. As we have seen so far, uncertainty has been mentioned basically in every slide. And this is because, okay, our definition of uncertainties is any type of limitation in the available knowledge affecting the range and probability of a possible answer to an assessment question. And as said before, this can be due to limitation in the evidence per se, that can be heterogeneous, can have different relevance, validity, or precision, or in the methods that we employ. We may have different assumptions, bring different uncertainties. We have different methods for collecting the data or selecting the studies or doing the analysis. So for all these reasons, the approach to uncertainty analysis is really very challenging to be defined upfront because it depends on several factors, including the availability of information, the established practices we have in the field, etc., etc. However, what is important to always keep in mind is that uncertainty is inherent in every step of the scientific assessment process, from problem formulation up to drawing conclusion, and should be considered, and ideally they should be minimized in every step of our assessment, and not just left at the end of the assessment process, because that could be a little bit too late and may also maybe too challenging. So I will now give the floor to Thor for the conclusive part of this info session.
Thank you very much. And now we are down to conclusions and recommendations before we will wrap up with the final question and answer session. And I think sort of the take-home message, well, based on what has been presented above, is that I think sort of it's my feeling at least that the new updated guidelines provides a harmonized framework for making protocols across EFSA's domains, and it draws on lessons learned during the sort of from the last decade of EFSA's work. I hope that we have convinced you that the problem formulation, the a priori paradigm, provides a structured approach for translating mandate terms of reference into scientifically answerable assessment questions and sub-questions. And we feel that this paradigm is adaptable and broadly applicable within and across various EFSA domains. And then I think it's expected to help to harmonize problem formulation and foster consistency in protocol development within EFSA. Now, and I mean, the reason why is that the a priori is a little bit more flexible than the sort of the PICO-PECO framework that is developed around evidence-based medicine. I mean, what EFSA is dealing with is much more diverse type of assessment questions than simply what is sort of formulated in a simple randomized controlled trial. What we have identified through our work is that it is important that the process of protocol development should be both flexible and proportional to the complexity and the resources allocated to each scientific assessment, meaning that, you know, the transparency and the expected gain in time by planning upfront should be, well, we should be able to harvest on that, but not overplan or use too much resources in the beginning and not having enough time to do the assessment. And I think that that's been highlighted in our work, and I think this is something that we have to sort of bear in mind. But I think that will come on a sort of it will not be an issue after sort of. And I think the years that we have been using protocols, there has been a lot of improvement in both efficiency and time taken. Now, what I have enjoyed during this working process is that, you know, we have been working on this across panels and across units in EFSA and sharing experience both with protocols, but also in terms of methodology and how we do things and plan things. And I think that type of understanding across disciplines, both in terms of terminology and methods, has been really helpful. And I think it is really needed in terms of improving and creating more robust scientific assessment where people from different backgrounds understand each other's languages and can see the similarities and differences across the work that we are doing in EFSA, which is quite diverse. Next slide, please. Now, what we come up as a recommendation in the guidance is that we have the sort of the theoretical document or the guidance document itself, and we have an annex which is supposed to help people to create protocol or sort of a framework for doing protocols where people can copy, paste, and delete certain parts of the annex and make it into a protocol. But what we realize is that these sort of standalone Word documents are a bit outdated and not really flexible all the time. So what we sort of would recommend is that we should have a sort of some kind of web-based tool that has a repository of protocols and an interactive sort of annex that would help people creating protocols in a more interactive manner where you can draw on existing experience. And I think in the long term that would help streamline the process of protocol development in EFSA. And also a sort of repository of existing protocols and Aprio examples will help and speed up the process when we have assessment questions that are challenging to plan. So that would be my recommendation and conclusion session. And now we are in the final round of Q&A questions, and we have a couple of questions. I thought we had one question that I would like to start with. We have a couple of questions from the previous session, but as far as I could see, we have one question here. Would you say that the C in the PICO is included in the Aprio model? I think it would be appropriate to take that question now. So whether the C in the PECO, the comparator, is included in the Aprio. And Laura, would you answer that question?
Yes, thank you for this question. I must say that we have discussed whether there was the need to include in the Aprio paradigm explicitly the C, that could be control, comparator, both meaning. And we had this discussion both with the contractor and within the working group. And if I recall well, the rationale for not considering them is that indeed it can have a bit a double twist, because it can represent the classical comparator when it comes to the efficacy question, and you have a sort of experiment in which, for instance, you apply a treatment to reduce the load of microorganism, for instance, and in that case you will have a traditional control that would be what happens if you apply no treatment at all. But it will have also a slightly different meaning when it comes really to what we define the intervention, because there it will be an alternative measure that you apply in real condition. And we thought that at the end it was in both cases implicitly covered by the Aprio, but indeed you might discuss whether instead we could come up in the future with including an additional element to cover this.
Thank you very much. We have don't see any new questions, but I encourage you to place questions into the chat box. But we have 4 outstanding questions from the previous sessions that we will, in the meantime, address. And there is a question from session 1. Are EFSA sister agencies adopting similar approaches on protocol development, I assume? Is alignment across NV agencies foreseen? And I would like to direct these questions to Claudia.
Yeah, thanks, Tor. Thanks a lot for this question. In fact, sister agencies have been made aware of the finalization of this draft document. We are now in the process of discussing with them, and it's important that we discuss now with them that the document is in a final form that allow us to illustrate the whole process, and as you have seen, to illustrate also practical application on the use of the guidance. So meetings with them are envisaged in the near future, and yeah, we are working together with our sister agencies. Thank you.
And there's another question on the PICO-PECO comparison, as far as I can see from session 2. What is the advantage of using Aprio over PICO-PECO? And I give the floor back to Laura.
Indeed, thank you also for this question that gives me the opportunity to clarify better why we are proposing a new approach, because indeed the PICO-PECO is there since years and it is applied in many fields. And it can work well in certain fields, such as the clinical setting. But it has also limitations when it comes to certain domains in EFSA, particularly because it doesn't cover the entire breadth of the type of mandates that we receive, particularly when it comes, think of the population, for instance. It applies well to the human population, the animal population. But if you think of the various examples that we provided before, if we think of the cell lines, organoids, but also the diagnostic test that we consider covered by the receptor concept that is extremely general and broad, this wouldn't fit well with the definition of population in the PICO-PECO. And also there were some missing elements in the PICO-PECO, particularly the pathway and the output. And the pathway is really necessary, particularly when it comes to the biological hazard, the plant and animal pest, where you need really to follow the evolution of the agent across along the food chain or the life of the plants and the animals. And without the specification of the pathway, it is really difficult to identify well the evidence needs and the method that you need to address the questions. And also the output. We believe that it is very important to specify since the beginning in the problem formulation which type of answer you want to provide, because you can answer to the same question in many different ways. And this, of course, again, has an impact and influence on the kind of evidence that you're going to retrieve. So in essence, the broadness of the APRIO compared to the PICO-PECO, and also the possibility to specify other elements that are relevant for some EFSA domains. Thank you.
And on the same question, I would like to call in Elisa.
Yes, thank you for the question. We acknowledge that APRIO represents quite a novelty in problem formulation, and although we are of course very confident that it will give advantages to problem formulation, this is really a part of the guidance where we would welcome feedback and input from external parties, especially those that are used to using PICO-PECO. It would be very useful for us to receive feedback. So it's just to highlight that the guidance is out there for public consultation, and we would really welcome comments on this part. Thank you.
And then we have a question. In the case of urgent advice, is it possible necessary to apply aspects from the protocol development process? And Elisa.
Thank you. Yeah, we did discuss this situation. Of course, in this case, it is not possible to dedicate much time to planning because we are in an urgent situation. In this case, the protocol would really represent a sort of very brief outline of the approach that is going to be applied together with possibly with the uncertainties that the urgency of the process will introduce. So in principle, yes, the principles are applicable. In terms of extensiveness of the protocol, it would be a very short document. and outline. Thank you.
Yeah, which is in line with what we say, that it has to be proportional to the time and resources, and that, I mean, I think we would follow that principle. Not seeing any more questions in the chat. We have one leftover question from session 1, which I will give the floor back to Elisa. Is there a way for people who are creating this question breakdown to make it available to others internally, perhaps, to view in the future? Sorry if out of scope, but this relates to the repository, and I think we can, I mean, within EFSA, such a repository would be open, but I mean, outside EFSA, yeah, please comment, Elisa.
Yeah, thank you for this question. It's not out of scope at all. It's related to the recommendation for future development that is outlined in the guidance and was described by Tor in his last presentation. Indeed, it is recommended to create not only a web-based tool for developing protocols, but also to integrate this tool into a repository of protocols, including, of course, the parts on problem formulation, in order to build on experience and share this experience as we progress in the implementation of protocols within EFSA. Of course, this accessibility of the repository would be first for internal use, for protocols that are not yet published, that are still in draft, but then why not, of course, opening it up also to external views when the protocols are final. So this is an important recommendation from the guidance that will be considered within EFSA for further implementation. Thank you.
Yeah, and not seeing any more questions, we have reached slide 117. We are a little bit ahead of schedule, which I think is a good thing. And if there are no further questions from anyone, then I think we are come to the stage where we wrap up. Next slide, please. And in terms of concluding remarks, I would maybe first ask if there is anyone, Elisa or Laura, that you want to come in and say before we sort of proceed with the. And if not, then let's go for the next slide. So then I would just like to thank you for attending this event here today. In case we did not manage to answer all your questions, you will find them published on the EFSA website, but I think we actually managed to answer all your questions, so that is a non-issue. The recording of today's event will be available on the EFSA website in few days, and we hope that you can find time to take a few minutes to fill out the evaluation survey that you will receive after this event, because, you know, such feedback, and it doesn't take a long time really, and helps us to improve our events, which some of you are attending maybe regularly, so I think everyone will benefit. And most importantly, we kindly ask you to provide any comments or recommendation on the draft guidance document through the public consultation. So the questions asked today are not taking into consideration there. So any comments that we welcome and are important for us should be submitted through the public consultation website, which closes on May 15, and you have a link there on the slide. So thank you very much, and I really enjoyed being here with you today. Thank you.